Okay, so our next speaker, continuing along the line of the immunology topic here, is also going to be talking about lipopolysaccharides, or LPS. Dr. Charay Jennings is completed her PhD at Stanford and is now going to be finishing the MD program very soon, like this spring. She's in the biology department at Stanford. And her title is going to be Calcineurin Inactivation Induces LPS Tolerance in Macrophages and Dendritic Cells and Protects Against LPS-Induced Toxicity in Vivo. Charay.
Good morning, I'm Charay Jennings, and I'm an M7, currently in the MD PhD program at Stanford, and I defended my PhD last May, and I'll be graduating in, 68 days, and, and I actually will be done with all of my medical school work in 48 days, so I'm very excited. My research was on the role of calcineurin and its deficiency in inducing LPS tolerance. And first I'm going to talk a little bit about innate immunity, but I've already heard some good stuff about it already. But innate immunity basically is your first line against an invader. So if there's a pathogen that's trying to infect the host, then the first line of attack is the innate immune system. And so you can do that through anatomic or physiologic barriers. And the next step would be to recognize what you're seeing. So you take these Toll-like receptors, which we already heard a little bit about in the last talk. And the third step would be to attack the invader, and that's using inflammatory and antimicrobial responses. So an example of a microbial component would be LPS. And some people often call LPS, they refer to it as endotoxin in general. So you'll hear me kind of interchangeably talking about it as LPS or endotoxin. And it comes from the cell surface of gram-negative bacteria and is recognized by Toll-like receptors. And briefly, this is a busy picture, but it's just showing all the different kinds of Toll-like receptors that have been identified and all the different ligands that they recognize. And then you can see that in response to these ligands, the Toll-like receptors lead to lots of different pro-inflammatory pathways. And calcineurin is a calcium and calmodulin activated phosphatase, which actually has a role in T cell activation. It's made up of two subunits, calcineurin A and calcineurin B, which is the regulatory subunit. And calcineurin inhibitors are very widely used immunosuppressants, and people use them every day in patients to help to prevent transplant rejection or an autoimmune disease or inflammatory skin diseases. And they're often used in conjunction with other drugs, but they're also limited by a lot of different toxicities, and most importantly, nephrotoxicity. And there has been a paper published that shows that the nephrotoxicity may be related to the activation of NF-kappa B, a transcription factor, very important in inflammation. So there was a postdoc in the lab prior to me joining the lab who did a lot of work in looking at the role of calcineurin in the Toll-like receptor pathway. And one of the things that he found was that here, this is the Toll-like receptor, and if you upregulate the expression of calcineurin in macrophages, then you actually inhibit the activation of all the downstream steps of the Toll-like receptor. And interestingly, if you add in calcineurin inhibitors, here FK506 and cyclosporin, or you block calcineurin using the siRNA, then you get activation of this pathway and then transcription of inflammatory genes, most importantly NF-kappa B. So my project was basically to look at what the physiological relevance was of this role of calcineurin and that we didn't know prior in the Toll-like receptor pathway. It's only present in macrophages and other innate immune cells, but not T cells or B cells. And so our hypothesis is basically that if you inhibit calcineurin activity in these innate cells and myeloid cells, then you will activate NF-kappa B and lead to a pro-inflammatory response. So the possible outcomes could be if you could either get an increased inflammatory state because of the NF-kappa B that's being produced, or you could get sensitization to further activation of TR ligands so that you get overreactivation. Or you could get desensitization, or LPS tolerance, which is commonly called. So LPS tolerance is often called endotoxin tolerance in the medical world, but it's usually a desensitization reaction to LPS. So once you get an initial exposure to LPS, you're unresponsive to a subsequent exposure. And it's thought that this is very important for limiting inflammatory damage due to excessive cytokine production, et cetera. And this can also be linked to development of sepsis. So tolerance is usually characterized by the downregulation of the inflammatory cytokines that are produced by NF-kappa B. And usually to determine whether tolerance has occurred, you just measure cytokines like TNF-alpha, IL-1, or IL-6. And in dendritic cells, it's more likely IL-12 that's going to be downregulated. So here is a picture showing here in the red box is the Toll-like receptor pathway. And all of these arrows are ways in which negative feedback tolerance occurs in this pathway. So all of the proposed mechanisms in tolerance are usually surrounding the Toll-like receptor pathway. And sepsis is considered to be a model for the dysregulation that happens in tolerance. And a lot of people believe that once you have survived sepsis, then you will become tolerant to subsequent exposures to bacteria, which can render you either what they call immunoparalyzed, meaning you cannot respond to subsequent pathogen infections, or you're protected from future septic events. And LPS comes in, and it can lead to the stimulation of all these different cytokines, which can lead to a lot of different bad medical sequelae. So you get coagulopathies, inflammation, and all of these bad outcomes. So the first model that I used was calcineurin inhibition, using the injection of calcineurin inhibitors, which I talked about earlier. And I just examined the in vitro and in vivo effects of these calcineurin inhibitors. And I used mostly FK506, which is one of the most used in the clinical realm, which is tacrolimus. And here is just a figure showing that if you pre-treat macrophages here, this is a macrophage cell line, and these are actually peritoneal macrophages from mice, and here is bone marrow-derived dendritic cells. And you pre-treat them with LPS or FK506, the calcineurin inhibitor, and then you come back with a second treatment of LPS to see if they will respond. Sorry, that's supposed to be micrograms. Then you see that if the cells that are pre-treated with LPS or FK506 are much less able to respond to that second dose of LPS, shown here in the black. And so we determined that maybe this FK506 is actually giving the same response as LPS and inducing this tolerant effect after an initial activation. So I looked at it in vivo and actually injected the mice with either LPS and their, the solvents, or FK506 and its solvent. And comparison, you see that those mice that were injected with LPS or FK506, the macrophages from those mice had a very impaired response to LPS. So, I mean, they had a very impaired response in vitro. So we took the cells from these mice that were injected and plated them in vitro and looked at their IL-12 and TNF production. And the cells from the LPS or FK506 induced mice produced much less IL-12 than their vehicle controls. The same here for TNF. And then I took a step further and looked at the NF-kappa B activation in these mice. So this is the vehicle or the FK506 injected mouse, and you see that in response to LPS, these macrophages are much less able to activate their NF-kappa B. So I looked even further by just looking at survival rates of FK506 injected mice once they were challenged with a lethal dose of LPS. And this would be equivalent to giving somebody endotoxin shock or sepsis. And these mice here were injected with LPS, these were injected with FK506, and these with the vehicle control. And you can see that the control mice did the worst. And the FK506 and LPS mice were protected from death from the LPS because of the tolerant response. So next I looked at whether these mice were not only protected from death, but protected from sickness as well. And I developed a recovery score, which is an arbitrary way to look at how sick the mice are, and I created a cutoff. So if they were able to run, we considered them completely well. So on this axis is actually all of the mice that received a score of 2 or above. And here you can see we start out with 100% of the mice being healthy, and then we go down to a very low percent of the mice being healthy. And here the bottom one is the vehicle control, and they do the worst out of all mice. So the LPS did the best, and then FK506 was protected from sickness. The second model that I used was a genetic model with calcineurin deletion, and I actually used the CRE-LOX system, where I used the a CRE recombinase under the control of a CD11B promoter, which is a myeloid cell-specific promoter, and that excised the calcineurin B1 subunit, which rendered calcineurin inactive in these mice only in the cells that were of myeloid origin, so macrophages and dendritic cells. And these mice were transgenic that I got from the Vacherie lab in Canada. And I don't need to go into all of those things, but so these calcineurin deficient macrophages from these transgenic mice had very much impaired cytokine responses. And here you can see the TNF response to LPS in the calcineurin deficient mice in the white bars is much impaired compared to their wild type controls. And the same for IL-12. Sorry, this is IL-12 here, and you can see that these calcineurin deficient mice seem tolerant to LPS. And if you look in vivo and compare the mice that have calcineurin deficient macrophages and those that are wild type, the calcineurin deficient macrophages survived a lethal dose of LPS a lot better than the wild type. And we think that this is due to the TNF production by the macrophages, because if you take the macrophages from these mice right before they're euthanized, then you see a much lower TNF production in the calcineurin deficient mice. So in summary, FK506 can induce inflammatory response, but then a subsequent LPS tolerant response in macrophages and dendritic cells, and that this can actually increase survival and recovery from a lethal LPS exposure. And that if you delete calcineurin in macrophages, then you can actually lead to a similar decreased inflammatory response and protection from lethal LPS. And we conclude that basically the reduction of calcineurin activity leads to this initial activation response, which was shown previously in the lab. And then my project shows that this also can lead to an induction of a negative feedback and tolerance response to subsequent LPS exposure. And then this can actually protect from the toxicity of LPS or sepsis. So this is important for determining whether there may be a therapeutic opportunity for patients that are suffering from sepsis, as well as further evaluating the effects of calcineurin inhibitors in patients, and they may be having further effects in the innate immune system as well as the adaptive immune system. And that's my daughter. And I just want to thank my lab and all the contributors to this project, Stanford Immunology, Department of Biological Sciences, the MSTP, and the Meyerhoff program. Sorry. Thank you. Questions? Thank you. Yeah, so it was just an arbitrary thing just to get a good sense of how healthy they were. We didn't measure, toxins in the bloodstream or anything like that, but we thought this was a good rough way to just see if they were able to return to function, because some of the mice didn't actually succumb to the lethal exposures of LPS. I mean, if we give them sublethal exposures, they recover. And we wanted to see if the mice could recover faster if they had this LPS tolerance response.
So there are a number of ligands that activate the TLR4 HMG1
Right.
S100 protein. So if you're tolerant to LPS,
Yeah, so there's been a couple papers looking at what they call heterotolerance to see if one ligand can tolerate to the other, and it seems to be a very complicated picture. And there's some that are and some that aren't tolerant to each other. So it depends, I guess. That's the quick answer to that.
Thank you.
Mhm.